bio people, we are in lesson 3 of genetics, and today we are going to talk about codominant traits and human blood types. Human blood types are an excellent example of codominant traits. Plus, who doesn't want to know about their blood type, right? So here we go. First of all, what is codominance? Codominance is when the expression of the alleles is equal for the phenotype. So remember, your phenotype is the expression of the allele. It's what you see when that allele is expressed. Is your seed green or yellow? Is your flower purple or white? Is the plant tall or short? These are all the expressions of the genes for these phenotypes. So here, for codominance, both of the alleles are expressed equally when you have the heterozygous condition. So a quick review: dominant versus recessive traits. You would have the red flower versus the white flower, and the red flower would win, and it would conceal the expression of this gene for the white flower. For incomplete dominance, we would have the red crossed with the white give us a pink. So incomplete dominance means they are still equal. There's no concealing. There is a blending instead. So we're blending the red and the white to get us pink, and about 50% of that red pigment shows up. Codominant is another time where the alleles have equal strength, but here, instead of blending, they're going to show up on their own separately. So if I had this red and this white flower combined and they were codominant, I would see red and white separately in the flower. This shows up in these fish over here too. So here we have the red fish, and here we have the blue fish. And when we do codominance, we use capital letters again, so it avoids confusion. Use the first letter of the phenotype you see. That's helpful. So red and blue, R and B. And then when they cross, you get this really funky striped fish who has both the red and the blue allele. So that's codominance. So how does that affect your blood? Let's see. So you have two basic alleles in your blood that create antigens that show up on your blood. So there are 3 alleles total. One provides you with an A antigen, one provides you with a B antigen, and the last one doesn't give you an antigen. We notate the A and the B antigens on the letter I. Often when we're doing genetics, they have this little carrier letter to help with tracing how things go together. And this indicates immunoglobulin, which is a class of antibodies that you find in your immune system. And they've aligned it with the type of blood you have, A or B or O. So A and B alleles are codominant. If you have both of these alleles, then you will have both of these antibodies in your blood. Antigens in your blood. What is an antigen? An antigen is a glycoprotein. So remember way back when we did cell membrane stuff? The cell membrane has these molecules that are embedded in it, right? These proteins. And then they have these carbohydrates attached to these proteins. These are called glycoproteins. They are basically a carbohydrate molecule attached to a protein. So glyco is from the sugar, right? That makes up the carbohydrate. And that creates an antigen on your red blood cell. Now, the interesting thing about antigens is you have these antigen markers on all of your cells, but they're different for every person. So the human race, which is everybody in Homo sapiens sapien, all have either A or B or no antigen on their red blood cell. Those antigens are the same from person to person. the antigens on your other cells in the rest of your body all have an antigen that basically signifies that that cell belongs to you. So all my antigens say Mrs. McCourt. All of Mrs. Sotomayor's antigens have her name on them. Your antigens have your name. This way, your body knows what cells belong and what cells don't. This is very important for your immune system because when a cell comes in that doesn't belong, the immune system goes on alert and attacks it. You want that because you don't want random cells coming into your body. It becomes a little complicated when you have something like a kidney transplant because the kidney coming into you is not going to have your antigens on it, and so your immune system is going to want to attack that new kidney. You need that new kidney, so your doctor would give you immunosuppressant drugs, which would suppress the white blood cells that attack the antigens so they don't attack the new kidney. So in your blood produces both where your body produces both of these antigens that are found on your blood. I is recessive to both. So the lowercase I represents basically no antigen. So you get an antigen A and an antigen B and no antigen. Okay, so what does this mean? Normally we talk about blood types and you will hear, I have type A blood, I have B positive, I have O blood. Those are your phenotypes. This is how we talk about blood with our doctor and with our friends and our parents, whatever. We just say, I have type A, type B, type AB, or type O. It goes a little deeper because there's a genotype involved. So if you have type A blood, that means that you have two alleles for blood type. So you can either have two A alleles or you can have one A allele and one O allele. So you would have type AA or type AO in your genotype. Both of these have the same type A phenotype. You're going to have type A antigens on the surface of your red blood cells, and you're going to have anti-B antibodies. Now, antibodies are what your immune system produces to attack antigens. So for every antigen you encounter, your body makes a new antibody to attack that specific antigen. Now, because blood type is the same for all humans, we already carry all of these antibodies for the wrong blood type. So if I have type A blood and someone tries to give me type B blood, my anti-B antibodies are going to attack that B blood. That's not great because when this attacks my B blood, it agglutinates the blood. So agglutination is clotting of the blood. The blood cells glob together around the antigens and basically kills the person with given the wrong blood type. So they basically generally have a blood clot to the brain and they die. If you have B blood, it's the same idea. You have two alleles. You can have two alleles that say B, or you can have one that says B and one that says O. You would have type B antigens on the outside of your red blood cell, and because you don't like A, you would have anti-A antibodies. So these antibodies are going to attack any A antigens that come in. So what about AB blood, Mrs. McCourt? Oh, let me tell you. So AB blood has two alleles, one allele for A and one allele for B. We have both type A and type B antigens on the surface of the red blood cells here. And we have no antibodies for blood type. We have other antibodies. But we don't want antibodies here because we'd be agglutinizing our own blood because there's only anti-A and anti-B. So if you put those into an AB blood, that's a problem. So the interesting thing about not having the antibodies is it makes this blood the universal recipient. That means they can take blood from the A people, the B people, the AB people, and the O people. So all of these people can give this AB person their blood. Now, O is special. O doesn't have the antigens that these guys do. So there are no antigens on the surface of the red blood cells. We use lowercase I here to represent this as sort of a recessive trait because these being codominant, they're going to equally show up. But if you have half of your alleles are an I, like they are up here for the Bs, we just say the blood type is B. We kind of ignore the O. Nobody goes around saying, I have BO blood type. Okay. This means this guy also has anti-A and anti-B antibodies. So he carries both sets of antibodies. So big trouble if the type O person is given AB blood, this will react, B blood, this will react, A blood, this will react. So the O person is not a recipient. They basically have to get blood from other O people. This makes them the universal donor, though, because they don't have any of these. They don't have any of these antigens to interact with the antibodies. You can give O blood to this guy, and this guy. Makes them very popular. Here's just a visual so you can sort of see what I'm talking about. You have, for a type A person, this person would be, we would say, I. Oh, this is not working as well as I wanted to. Okay, so all the spots on this person are taken up with an A. So their genotype would be IA. If a few of these were blank, then we could say they were IAI. This is a B person, so they're going to have IB. And again, if some of these were not present, that would make him or her an IBI. This is type O. Type O has no antigens, so they look like that with the genotype. And then this is your AB guy. This person has no choice. They are going to be IA, IB. No option of the O blood type showing up at all. But wait, there's more. You have an RH factor. It's one more marker on your blood, one more molecule that you got to worry about. It's RH is it's called the rhesus factor because it was originally discovered in rhesus monkeys. If you are Rh positive, we again have a 2 allele system because that's how we roll. This 2 allele system can be Rh plus, which means we have 2 Rh factor genes here, or alleles, sorry. Here we could be Rh plus, Rh negative, so I have 1 Rh allele, and then I don't have a second one. The second one is not Rh allele. And then Rh negative means that these are not producing the Rh factor. So this adds into our blood typing to give you that AB positive, O negative factor. So here you can see on these examples, your A antigen is that golden triangle, and the B antigen is the blue triangle, and then the Rh factor are little black squares. So this person has got everything. This is your universal recipient. I can give this person Rh factor, no Rh factor, A blood, B blood, A positive, A negative, B positive, B negative, AB positive, AB negative, O positive, O negative. He could take it all. A positive means that they have the Rh factor, so they can take A blood and they can take O blood. They can take O positive blood because this Rh factor is in both of them. They can take O negative because there's no Rh factor to interfere with what's already going on. With B positive, they can take B blood, O positive blood, B negative blood, and O negative blood. Okay? Essentially, if you're positive, giving you any of this negative blood or I shouldn't say any, but blood that has a negative factor is not going to cause a reaction. But if you are B plus, if you're B positive and you try to give your blood to this B negative person, that Rh factor is going to cause an antibody reaction, and you will cause clotting and death. So you have to be careful with the Rh factor as well as the A and the B factors. This O negative is the universal donor. There's no B here, there's no A here, there's no Rh factor. This guy has no antigens to attract any attention in anybody else's blood, so you can give O negative to everyone. Luckily, there are a bunch of O negative people, and because most people are actually Rh positive, the O positive people can help out a lot with the donations, because there are actually a lot more O positive people than O negative people. But if they find out that you're an O negative blood type, they're going to be calling you for donations. Okay, so this just gives you another little description so you can see what's going on with the A's and the B's and the minuses. And I just liked it because it was fun. So what else is going on? You also have some gene expression that is determined by more than one gene. We call it polygenic inheritance, and it kind of gives a reaction on a sliding scale. So basically, this curve shows how skin color changes based on the number of alleles that are activated. So these, because this is for skin color, the activation is of melanin in your skin. So the more melanin activated, the darker your skin's going to be. This shows that there's a peak in the middle. There's fewer people that really, really light colored and fewer people that are really, really dark colored. And then in between, there's a complete range. If you look around the room, you're going to see everybody has a slightly different skin tone. There's really nobody that has exactly the same skin tone. This analysis can also be applied to height, weight, intelligence, and some behaviors. So this one is looking at 3 genes that are affecting skin color, and each of them have 2 alleles. So you're looking at 6 different alleles that will affect your skin. So essentially, it's just so you know that it's not always a straight up, you have the allele or you don't. Finally, the environment can also affect how your genes are expressed, what your phenotype looks like. So the Arctic fox has heat-sensitive alleles that know when it's hot and when it's cold. So when it's cold, our fox, his fur turns white so he can blend into the snow. And when it's warm in the summer, he gets brown so he can blend into the grasses that he lives in. And here's just a picture of how the melanin pigment is sent into the hair follicles, and that gives the color to the hair. You also have melanin in your skin. Melanin is a big deal. It's all over the place. Melanin in your skin will be activated by exposure to the sun. So this woman doesn't look this tan in the wintertime, but she spent a lot of time in the sun, and so she looks tan. And finally, pH can also affect the expression of alleles. This is a hydrangea plant, and blue ones are actually in like an acidic pH, and pink ones grow when you have an alkaline pH. So it's actually the opposite of pH paper, because pH paper, blue is a base, but the blue hydrangea actually indicates an acid and the pink indicates a base. And that's about it for today. We have one more lesson coming, and hope you've enjoyed it, and thanks for listening, and let us know if you have any questions.